Um, it's kind of hard to believe that anything is going to bloom, but we're going to talk about phytoplankton today. And one of the big phenomena in the world is the spring bloom. So the first thing I want to do is ask if anybody has any questions about anything. So for the practicum, you know that it's on the website. It's week 6, and so if you look at it, it will tell you what concentration of the concentrated solution you're going to make and what concentration of the dilute solution. So when you walk into the practicum room, you should be ready to go. So it's all there. If you walk in and you start doing the calculations, and the next student comes in, we'll leave you half an hour for the balances. But if at the end of your half hour, where you have exclusive use of the balances, because we'll do 4 people every 30 minutes, then you're just going to have to wait. So make sure you come in ready to go. Okay, no questions for me? I just want to make a few comments on the homework, and then today's lecture and lab are the relationship between nutrients and phytoplankton. And we'll go back to the stoichiometry concept and how you can use that to predict biomass, and then talk a little bit about how low nutrient concentrations can limit the growth rate. And first I'm going to go over what's a phytoplankton, what's biomass, and then microbial growth rate, which is what the lab is all about. Next week, make sure you bring your goggles because you'll be doing the analyses from the samples that you're going to collect today. This is the calculated absorbance from the tank data. And you notice it starts going non-linear. And this is the absorbance from the spectrophotometer. And you notice that it's really linear. You'll also notice that the units for the first few samples are very, very close. This is calculated from the tank light data, and this is recorded directly from the spectrophotometer. And the reason is, if you look at the absorbance spectra of the green dye, remember it's green because there's a blue dye that McCormick's adds to the food coloring that you buy, and there's a dye that absorbs in the red. And so the combination of a blue absorbing and a red absorbing dye give you the green that you see because it's not absorbed. So the spectrophotometer has a 1 nanometer resolution. And so it's really monochromatic light. And the light in the tank is broad spectrum. So we're measuring in the spectrophotometer, it's at 630 nanometer. So it's just one wavelength of light, whereas it's a broader spectrum than the spectrophotometer. And so there are wavelengths that are not absorbed as well. And this is really actually similar to light in the ocean. Who dives? Anybody dive? Okay, so when you go down about 30 feet and you're wearing bright red gloves, what color are they? They're black.
They're not as bright.
Yeah, because red light is absorbed preferentially in the ocean. And by the time you get to about 30 feet, 10 meters, there's no red light. And so it's a combination. So if you were to go diving in the ocean and bring a color wheel with you, what you would see, so depth in meters, is the attenuation of red light would look like this. So this is the intensity of light. Red light doesn't penetrate very deeply. If you're in the coastal zone, green light penetrates more deeply, and blue light looks like this. If you were to go diving off of Hawaii, what you would see, because it's very, very clear water, it's nutrient poor water, and so there's no phytoplankton, and it's far from the coast, so there's no colored dissolved organic matter. What you'd see is red light looks just the same in attenuation. Green light would look like this, and blue light would look like this, penetrating down to 100 meters. When I was in graduate school, you used to be able to go diving off of a research vessel. And let me tell you, when you've got 5,000 meters of water below you and you dive in and you look up at the ship in this crystal clear blue water, it's absolutely incredible. And so in the ocean, there is a different attenuation of different wavelengths of light. And it's actually really similar to what happens with this dye. So if you were to have an ocean that was colored with this dye, you would have green light penetrating very deeply. And if you were to just measure a broadband spectra, you would get, so like if we just took white light and took the attenuation spectra of white light, it would look like a combination of the two. So the point of all of this is, and I am getting your homeworks back in the next few days. I put the answer sheet so you wouldn't lack for some kind of feedback. But one of the big differences between the tank and the spectrophotometer is it's monochromatic light. And so I want you to remember that when you have monochromatic light, Beer's law really works. So, you know, don't get fooled by the tank. The other thing that I noticed in the homework, has chemistry started calling molecules particles? Everybody talked about particles of dye. If you have scattering particles in the spectrophotometer, you violate Beer's law. Dye molecules, the dye is a solution of molecules and they're not particles. And I think like 85% of the people talked about particles. It's dye molecules. So I felt like I was going through another Niagara Falls moment by the end. So I'm about 60% through with the grading. If you're having any issues at all with the graph, please see us. It's going to be a short lab, and so this would be a great time. And a number of people plotted. If you notice the scale here, it's not linear. And so Excel is stupid. It's deaf, dumb, and blind. You have to do the driving, so make sure you pick what's called a marked scatter plot. And so if you see something that looks nonlinear and you're supposed to be plotting linear volume or whatever on the x-axis, you've picked the wrong plot. So make sure that you don't do that. The other thing is I realized that homework question 4, this is not homework 4, this is homework 3, question 4, that there was it was a poorly worded statement. I got halfway through and I realized that it was ambiguous, so everybody's going to get full credit for question 4 because it was my fault. Couldn't figure out. Some of you were plotting data from the applets, and this is not what I intended. I meant to take a look at the applet in terms of the concept. Figure captions, please read this. I'm not going to read this through, but this is on the website in the lecture in terms of pointers for figure captions. There is, if you work for the federal government, you have to write a visually impaired version that would allow somebody who is completely visually impaired to have the figure caption read to them, and everything is read electronically now, that would completely describe things. Most journals don't have you describe what the function is in gory detail, but you should be aware if you work for the federal government, you may have to be able to write more detailed descriptions that describe the plot in terms of accessibility. So anyway, take a look at the figure caption on there. So today what I want to do is we're going to work with phytoplankton, Thalassia pseudonana in today's lab, and you'll each have a chance to come during the week and do sampling for this experiment. So first, although I think everybody here has taken Dr. Townsend's class, I just want to point out a few relevant aspects of phytoplankton. Biomass, notice that mass, remember the unit of mass is a kilogram, and so we measure phytoplankton abundance and we call it biomass because it's living mass. And what you'll do in this experiment is use chlorophyll, which is a pigment, and it can be measured in units of milligrams per meter cubed in the ocean. We're going to use that as a proxy. And what a proxy means, it's something that represents something. So if you've ever read any papers about plankton, if you take Dr. Runge's zooplankton and ichthyology class at the Darling Center, they're really interested in the food for zooplankton and ichthyoplankton. So they'll be measuring chlorophyll as an index of the phytoplankton biomass available for grazing. So we'll talk about that. And then growth rate. Microbes typically grow by binary fission, and you're going to be measuring the growth rate, which you'll be calculating in Excel. So that's why it's really critical that if there's any issues with graphing, you get that straightened out. And then there are three things that the experiment is supposed to do in terms of showing the relationship between phytoplankton biomass and nutrients. And the question is, as phytoplankton grow, what happens to the nutrients? So nutrients, and we're going to be measuring phosphate. And the reason we do phosphate rather than nitrogen is because you can measure phosphate by adding that one reagent that you did last week. Nitrate you have to reduce to nitrite, and you do that on a cadmium reduction column, and it just is a lot slower, and it's not as fast. So we're going to measure phosphate because it is an important nutrient in the ocean, and because it's easy to measure. So the question is, do they change the nutrient concentrations? Can nutrients set an upper limit to biomass? Last week I talked really, really briefly about the Redfield ratio, and that's the molar, and molar is a really important part of this, the molar relationship between carbon, nitrogen, and phosphorus. And has anybody ever seen a polluted body of water where a lot of really disgusting green, orangey phytoplankton? So if you have pollution where there is an excess of nitrogen and phosphorus that are added to the water body, you can use this Redfield ratio to predict what the upper limit of phytoplankton could be. And that could be important in terms of trying to figure out, do you really have a problem in management, or is it not going to be so bad? So it's a really important tool in terms of water quality, as well as trying to figure out what goes on in the open ocean. And then the third question is, do low nutrients limit growth rate? So there are two things that nutrients can do. They can set an upper limit on biomass, and when they're low, they can control the growth rate. And the function of the growth rate, if you've taken enzymology, looks very similar to something called the Michaelis-Menten enzyme kinetic. So if this is the concentration of the nutrient, the growth rate is going to have a function that looks something like this. So when nutrients are low, they can downregulate growth rate. When they're high, growth rate isn't affected by the nutrient concentration, but it can limit the biomass. So be clear in the distinction between the two controls. One is on mass and one is on rate. And rate has a unit of time. Mass does not have a unit of time. So here's some phytoplankton. This is about 1 micron Synechococcus. You tend to find this in the open ocean where nutrients are very low. This is another cyanobacteria. So these two, Synechococcus and Prochlorococcus, are what are prokaryotes. They're photosynthetic. Here's a pennate diatom. This is our friend this week, Thalassia syra westflogii. The genus name is always capitalized. The species name is never capitalized. So if you go on a job interview and you want to prove you don't know anything, you capitalize both of these or you capitalize nothing. If you want to show you know something, make sure the genus is capitalized and the species name is not. The guy we're going to use today is a small centric diatom and has an upper and a lower valve, and this is called the girdle. The frustule is silica, and that's one of the things about diatoms is they have a silica frustule. And it's about 5 microns in diameter. So it's small, it stays in suspension. Diatoms also form chains. Here's a dinoflagellate, serratium. Dinoflagellates in the Gulf of Maine, the Alexandrium species, which are these two, create all kinds of economic problems because they have paralytic shellfish poisoning. Coccolithophores, which produce something called dimethyl sulfide, that causes cloud nucleation, and so there's an interaction in terms of the climate. So anyway, these are just some pictures of some of the phytoplankton, and the important thing is that they're photosynthetic. So that means they're going to use inorganic nutrients, nitrogen and phosphorus, and they're going to use carbon dioxide as the raw building blocks. And they're going to use the sun as a source of energy. So they're basically little photosynthetic factories. And there's all kinds of work going on trying to figure out how do you create photosynthesis on an industrial scale. So there are a lot of people very interested in the photosynthetic machinery that allows phytoplankton to do this. And CO2 has an important aspect in terms of climate, because of the ability of phytoplankton to take CO2 and turn it into an organic compound. So here's the basic equation for photosynthesis. Respiration, of course, goes in the other direction. So they're photosynthetic, meaning they use inorganic nutrients. We would never make it if we had to use inorganic nutrients. We depend on photosynthetic organisms to make organic carbon. And they have to capture the sunlight somehow, and they capture it by photosynthetic pigments, and one of the prime ones is chlorophyll. And so we can take advantage of the fact that chlorophyll is only in photosynthetic organisms. And so we can therefore, that's why it's so beautiful as a proxy. You know, if I go look at a fish, it's not going to have chlorophyll. So I can take advantage of the fact that chlorophyll is only in photosynthetic organisms, phytoplankton in the ocean, and I can exploit that and use this as a proxy for measuring the phytoplankton concentration. So why should you care? Every breath you take has the probability that some of the phytoplankton produce that oxygen. There's oxygen. It's a byproduct of photosynthesis. In fact, phytoplankton are responsible for about half of the oxygen produced on the planet. So next time you take a deep breath, take a deep breath. Take a phytoplankton, because they're responsible for about half of it. They fix about half of the carbon on the planet. This is a petagram. Notice it's a capital, so it's not a picogram, and that's 10 to the 15 grams. And so there's an uncertainty in that estimate, and a lot of people like Dr. Thomas, myself, Dr. Boss are really interested in trying to reduce the uncertainty in that number. So phytoplankton fix carbon, and that allows them to be the base of almost all aquatic food webs. And it's important in terms of the carbon cycle. And some people think that seawater is slowing down. So there's two ways that carbon get out of the atmosphere into the ocean. One is through chemical equilibria, and people think that that equilibria is slowing down. And the other one is through photosynthesis and then sinking of phytoplankton to the deep sea. So they play an important role in the carbon cycle. And so hopefully you care about phytoplankton, And now a key question that many of us ask is, actually that should be an E, what affects the phytoplankton biomass, their growth rate, and their distribution? And with the changing ocean conditions, is that going to change? And science, if you can't, at the end of the day, predict something, then you're really not advancing science. So it's about prediction. And so knowing what controls their growth is going to help us predict what happens in a future ocean. And maybe that future is getting, I was just in Montana, it was 80 degrees over the weekend. 80 degrees in April, unbelievable. So here's mass. That's the amount of phytoplankton, and we can express it in terms of grams of chlorophyll per unit volume, carbon, nitrogen, phosphorus. And so the units are going to be probably milligrams per meter cubed. Now, just remember that chlorophyll, this is, you want to get it to an SI unit, but it's a little bit complicated because we're not in the lab, we're not going to measure chlorophyll directly. And so just keep that in mind, and I want to touch on why you are going to be challenged in terms of going from what we're going to measure in the lab to chlorophyll as an SI unit. So biomass is mass of material. Growth rate is a rate. And we're going to measure growth rate as doublings per day, how the cells divide and then increase in number. And just remember that the rate is a function of nutrient concentration. And so one of the questions is, as nutrient concentration changes in your culture, are you going to see a slowdown in the growth rate? It's a function of temperature. So at colder temperatures, phytoplankton grows slower. It's a function of light. In the culture box, we're going to try to keep the light constant. And so you're going to mark where your culture goes so that you can keep the light constant. And it's a function of pH. So keep in mind mass versus rate. So here's the chlorophyll molecule, the chlorophyll absorption spectra, and chloroplasts in what will become our good friend Thalassia syra. So we're looking at it from the top down, and all these little tiny things are tiny chloroplasts. So here's the molecule chlorophyll. It absorbs in the blue and the red, and we take advantage of that in order to measure chlorophyll fluorescence. So remember, we have something called the Serre absorption band and the Q absorption band for chlorophyll. There is, just like the Redfield ratio, a relationship between chlorophyll, carbon, nitrogen, and phosphorus. But if you wanted life to be easy, forget it. There are some caveats. I don't want you walking out of here and saying that I can always do a direct chlorophyll to carbon. Under controlled conditions, we can use chlorophyll to predict carbon, but there's physiology, and many people have studied how the carbon to chlorophyll ratio changes. So the big picture is there's a relationship. The fine print is it's more complicated, and if you have 20 hours, I'd be happy to sit down and talk to you about it, because it's really complicated. And it's fun. It's a puzzle. So here's something that we're going to use in the laboratory to measure the concentration of phytoplankton, and that's chlorophyll fluorescence. If I take an extract of chlorophyll, so here's a green tube, it looks green. Just pass this around. What you're going to see is something that looks blood red, and the phytoplankton, the chlorophyll molecule is absorbing light, and it's re-emitting a fraction of that as red light, as chlorophyll fluorescence. So you can see that in a test tube extracted chlorophyll. That's acetone extract. And then this is a chain of thalassemia. Each one of the spikes is a phytoplankton cell. And if you look under a microscope that's set up with a blue light and a filter and a red light, a combination of filters, you can actually see these cells glowing in the dark. And they're emitting that red light, similar to the what's in that test tube. So on the previous slide, I said there's a relationship between phytoplankton biomass and the chlorophyll concentration. In actuality, it's a range, and I'm just showing it in terms of the general principle. And if you go on a cruise, you do a lot of calibration in order to tie this down for wherever it is you're working. So here's a logic statement. If biomass is proportional to chlorophyll, and if the emission of red light, fluorescence, is proportional to chlorophyll, then biomass is proportional to fluorescence. So we've got an if-then statement. And so the argument is that when you control the measurements, you can exploit these relationships to use fluorescence to get at biomass. And that's what you're going to do this week. And I'll keep the lights out just so that you can see that, that fluorescence. So you're going to be measuring that this week. And so the term fluorescence comes from feldspar and Stokes of Stokes' law. Did you do Stokes' law in the lab last time? No Stokes settling? Okay, he was, you know, these people 200 years ago, 100 years ago, really, really smart. He figured out settling rate is a function of size, and he also figured out that if you shined UV light on feldspar, that you could get a purple light that was emitted. And the term that you may hear, like if you take analytical chemistry, is something called Stokes shift. And that is if the absorption spectra looks like this, the light that's emitted will always be at a longer wavelength because low wavelengths are higher energy and short wavelengths are higher energy and longer wavelengths are lower energy. And so energy can only go downhill. And so this shift to a longer wavelength is called a Stokes shift. And if you remember, that absorption was an electronic transition, moving an electron from the ground state to a higher state. Well, fluorescence is the emission of some of that light. So you have a loss of energy as fluorescence. And not all molecules are capable of doing this. Most molecules are capable of absorbing light, but most of them get rid of that energy by radiating heat, by releasing heat. So there's only a small subset of molecules that will emit light. So here's the chlorophyll absorption spectra. Absorption in the blue, the so-called Sorey band, and then absorption in the red, the so-called Q band. And if you look carefully, there's a shift in the emission, and so light is absorbed, can be absorbed by blue or red, but that energy is transferred so that there's only emission at that Q band. Now, the important thing to take home from this is we can shine a blue light on our phytoplankton, and we can measure the red light. If we shone a red light on our phytoplankton, we would have a really hard time distinguishing between the red excitation light and the red emission light. So we're taking advantage of a really cool property of chlorophyll. Now, if you remember in the spectrophotometer, we had the light source, we had the collimator, we had the prism, we had the sample, we had the detector. And so everything went in a straight line. You remember how you set up the tank? Well, with fluorescence, it's orthogonal. So we have the light, and then we have a filter that allows blue light to pass. It kicks out any red light. So it only allows, it's called a short pass filter. So it blocks out wavelengths longer than about 470. So we have the light passing through a filter, detecting the sample, and then fluorescence is isotropic. And so what isotropic means is the light is emitted in all directions. It doesn't have any direction. So you could measure the emitted light, from any direction. But what you do is you do it to a right angle because that minimizes, filters are not 100% perfect. So it minimizes any red light that might have snuck past that filter, possibly getting into your detector. So you take advantage of this right angle geography, and then you have a red, what they call a long pass filter that only lets red light get to the detector. So you excite with blue, the fluorescence is emitted in all directions, and that's what this is supposed to be. So you've got your light source, it only lets one color through, your sample, which emits fluorescence in all directions, and then you pass it through another filter to clean up the light, and you have a detector. So just keep in mind that it's really important if you wind up going to work in a lab that you understand your instruments and you understand your equipment and you understand what the principles are. And so this is right angle geometry. So here's hemiolus, a diatom, and there are a bunch of chloroplasts here. So this is a cell that has a lot of empty space. And if you put it underneath a microscope that has the blue excitation light and that red blocking filter, this is what you'd see, which is actually pretty cool. Now, the spelling is fluorescence. Please don't let me see any fluorescence. Go work with King Arthur if you want to work with flower, but fluorescence, it's UO, not flower. Where's the tube? Is it still? So we're almost at the end. I'll keep the lights out until everybody takes a look at it. Okay, so what's a phytoplankton? How do we measure? What I want to do is jump to this relationship between the biomass and the nutrients. So here's Albert Redfield, who worked at Woods Hole Oceanographic Institution, and he looked at inorganic nitrogen in deep water, and he looked at the composition of phytoplankton, and he found that there was about a 16-to-1 ratio on a mole-to-mole basis. So 16 molecules of nitrogen to 1 molecule of phosphorus. And then if you add carbon into that, he found that worldwide that this ratio of carbon to nitrogen to phosphorus was 106 to 16 to 1. That is something I would like you to remember, because I will ask you if you had 1 mole of phosphorus, how many moles of carbon could a phytoplankton generate? And the answer would be 106. So this is something that I would like you to remember. And so this is the stoichiometry, the Redfield ratio, that allows you to predict the upper limit of phytoplankton. I am a person that likes boundary conditions. I want to know what the upper limit is and what the lower limit is. And then the fun part is trying to figure out what it actually is. So it allows you to predict the upper limit. So are we good to turn on the lights? So in general, this would be, reality would be in here someplace because there could be other things affecting it, but this is the general relationship. More nutrients give you the possibility of having more phytoplankton. The relationships are always ugly. Here are some global averages taken from many different places all over the world. Notice they're log-log scales because this prediction of red field gives you an upper limit, but there's a trend. More nitrogen, more biomass. More phosphorus, in this case, it's using chlorophyll as a biomass. In general, in the ocean, phytoplankton are limited either by nitrogen or iron. In freshwater systems, either by CO2 or by phosphorus, although there are places in the ocean where phosphorus can be limiting. If we look, could you just flick that next? Thanks. So this is a satellite image of the equatorial Pacific. And this is during an El Nino year. And during an El Nino year, fewer nutrients come up to the surface. During a La Nina year, more nutrients come up from the surface. So this is a great visualization on an ocean basin scale that nutrients do affect the concentration of biomass. And if you wind up being able in your senior year to take Dr. Thomas's satellite oceanography, and actually if you take MATLAB, you'll probably get some exposure to some of these data sets. You can use them to ask questions about what's happening in the ocean. But this light blue says low phytoplankton, this greenish says there's a lot of phytoplankton. And in terms of, oops, sorry, one more time. Chesapeake Bay, this is the tidal Potomac, and this is a bloom of a little phytoplankton called microcystis. And so it was a 20-meter bloom. If I were you, I wouldn't even want to go boating in there. So this was about 30 years ago, and they're doing a better job in terms of wastewater treatment. But there's actually still a lot of pollution in the Chesapeake Bay. But anyway, that is one of the consequences of too much phytoplankton. So how do phytoplankton grow? They grow by binary fission. A lot of phytoplankton have sex, but they don't use it for reproduction. They use it for other things, like diatoms use it to reset the cell size. So you start out with what they call a mother cell, and when it gets big enough, it divides into two daughter cells. And at your leisure, just take a look at the relationship in terms of R cubed and volume. So the mother isn't that much smaller in diameter, but it is, because it's divided in two, about half the volume. So they divide in two. That's why we want to use, for growth rate, and we're going to call growth rate K, we're going to use doublings per day because the phytoplankton divide in half. So there are other ways that you can represent growth rate, but that's the way that we're going to do it. So here's a diatom cell just getting ready to divide. And so that leads to a daughter, leads to another daughter. And so you get division, and then these cells also divide. And just as a really simple cartoon, here you got one cell, two cells go to four cells go to 8 cells, assuming that they're all on the same track. And guess what? You can use a positive exponential to represent this increase in the biomass. And the growth rate lets you predict what your biomass is going to be over time. So here's a growth rate where they double every 2 days. Here's a growth rate where they double every day. And so you can see that a difference in the growth rate can, if all things are constant, lead very quickly to a winner and a loser in terms of total biomass. So you're going to use, instead of number of cells, you're going to use fluorescence as a way to hopefully your cells will grow in your culture, and hopefully you'll see an exponential increase in the fluorescence. And you're going to, just like you did for the log transformation of transmission, you're going to do a log transformation of this increase in fluorescence versus time, and the slope of this log transform data is your growth rate. That will give you directly the doublings per day. And so you'll make those measurements as part of your homework in 2 weeks. So you're going to find exponentials everywhere. It's like when you start feeling like you have a little bit of pneumonia and you have an exponential microbial growth rate, in a day or two, you may have a lot of pneumonia. So you find this everywhere. Okay, your growth experiment that you're going to do, people do this in research labs. Maybe you wind up working for Dr. Lee Carbas or Dr. Susan Brawley. They often, as part of their work, measure growth rate. So here's a paper that was published in 2010, and it looks at growth rate as a function of pH. So every data point that you see on that curve was derived by doing a different experiment at a different pH, calculating the slope, which is the growth rate, and then plotting it to see, does pH, which is changing in the ocean, does it matter? Does it affect growth rate? So you might, if you look at this generic growth rate as a function of something, here's pH, and it says low pH for marine phytoplankton, they don't do so good. This is some data from Dr. Boss's lab, and she was looking at alexandrium, which is that toxic dinoflagellate that causes all kinds of economic and health problems in the Gulf of Maine. And each one of these, let's see, no, this is cells per ml. So this is very similar to what you hopefully will see, is that you get this exponential increase, and then part of the question is, does that happen forever? When does it happen? When do you run out of nutrients? When do you start seeing a decrease? So this is called a growth curve, and it has sort of an S-shaped function. So these are, the experiment you're going to do is really similar to what a lot of people do for their research questions. So today's lab, there's 3 questions. How does phytoplankton biomass change as a function of time? So that's for calculating the growth rate. Does the initial concentration affect the final biomass? So that's setting this upper limit. And you're going to do a variety of different concentrations. So different groups will do different concentrations. And then does the growth rate change as a function of phosphate concentration? Do you just keep going exponential forever, or do you start to see tailing off? And these are questions that your data hopefully should provide. So there's going to be different groups will have different concentrations of phosphate, so you'll be adding that. It's the Lasiaceira weisflogii. We're going to keep the temperature constant, and if you put the flask back in the same place every time, we should keep the light constant. They're about 5 microns, so smaller than a human hair. We spent a lot of time talking about instrumentation. So the sampling today, you're going to set it up today and sample, and then we need somebody from each group to come in Tuesday, Wednesday, Thursday, Friday. You'll finish the sampling on Monday. The Tuesday group will start today, and then you'll come in Wednesday, Thursday, Friday, and next Monday, and then finish the sampling in the lab. So you're going to be measuring the fluorescence in this instrument. Do not touch. We've got this set up. You're going to be using the fluorescence as a proxy, and then you're going to filter the sample, get rid of the cells, and then you're going to freeze it and measure the phosphate next week. I'm going to go over in lab. There are some really great ways that you can screw up, so I want you to think. If you know that the person last week measured a relative fluorescence unit of 100 and you get 0, you did something. And I'll show you what you can do. I want you to think. And then you're going to use standards. We have a different kind of a standard for the fluorometer. You're going to freeze your sample, and then next week you're going to measure standard curve, and then for your homework you're going to calculate the phosphate concentration in your culture. Okay, so see you in lab today or tomorrow.